All right, I think we can get started. So good afternoon, good night, or good morning, wherever you are. Welcome to Speaking of Oats. My name is Pablo Olivera. I'm director of Oat Global. Speaking of Oats, it's a partnership between the Oat newsletter and Oat Global. So Charlene White is editor of the Oat newsletter, is with us today, and she will handle the question and answer after the seminar. We are glad you are here today. And before I introduce today's speakers, I want to take a moment to remind everyone that you can use the Q&A widget to ask questions throughout the presentations. Please use the Q&A widget and not the chat function for questions, since this preserves the question for later access. Today's session is being recorded and will be made available later via YouTube. So today's speaker is Duong Nguyen, a postdoctoral researcher at Melania Figueroa's group at CSIRO, Australia. Duong will talk about oat crown rust today, and the title of his seminar is Leveraging Molecular Tools and Data Integration to Solve the Oat Crown Rust Puzzle. Thank you so much, and now the floor is yours, so you can start sharing the screen.
Thank you, Pablo. All right, thank you, everyone. So yeah, just want to confirm if you see the screen here. Yeah, and yeah, thank everyone for joining this online seminar. And speaking of oat, and thank Charlie and Pablo for organizing this seminar and giving me the opportunity to talk to the oat community. And today's so first introduce my name is Nguyen, and I'm currently working at CSIRO in the Pest and Disease group. And today I'm going to talk about how to leverage molecular tools and integrate data to solve the oat crown rust puzzle. And just for the first slide, I would like to introduce about the team member working at CSIRO in the project, Durable Resistant to Oat Crown Rust. And this project is funded by CIDC with long-term aim to deliver the durable resistant to crown rust in oat. And in this project, we are working on both the host and the pathogen aspect. So we have PhD student Eva Henningsen here working on the pathogen, try to study the evolution of the PCA population in Australia. And I have myself here working on the host aspect, try to develop molecular tool to improve resistance in the oat crowns and in the oats, and try to identify the source of resistance. And some briefly information about the current oat production in the world and the impact of crown rust to the different oat growing region. So nowadays, oat is grown in more than 80 countries with the total production of more than 22 million tons. And the top 3 countries growing most of oats are Canada, Russia, and Australia. And in Australia, we would like to improve the production due to the global demand. And here in the map, you can see the color coding representing like the impact of crown rust to the different oat growing region. So the darker color indicate that the more widespread of the incident. And here you can see that Canada is the place suffer the most of the impact from crown rust. And in some place where the susceptible cultivar is grown, the yield loss can be more than 50%. And the researcher and the breeder have proposed that genetic resistance to crown rust is the most effective and efficient method for the disease management. And talking about the genetic resistance, so far in the literature, we have got more than 100 resistant gene assigned to the oat genome, which include both the race-specific and non-race-specific type. However, none of these were cloned yet. And in this presentation, I will refer to the crown rust, Yersinia crotata avena as the PCA for the short acronym. And The key concept about the race-specific resistant is based on the gene-for-gene model, and for each resistant gene in the plant, there will be a corresponding virulent gene in the pathogen. So if the host receptor recognizes the rust effector from the pathogen, that will trigger the immunity system in the plant, lead to the resistance. However, if the host receptor doesn't recognize the rust effector from the pathogen, that will lead to the infection of the pathogen to the plant. And as you know that the use of the race-specific for disease resistant has been around in the last decade and been very popular. However, there is an issue with the use of this kind of major gene resistant because the virulent tend to appear to the major gene for about 5 years or less after deployment in the field. So in this project, we would like to figure out the question how to deliver long-lasting rust resistance in the field. And to do that, we have two different approaches. The first one is to study and make use of the adult plant resistance. So the adult plant resistance seem to be more durable and provide broader spectrum of resistance and offer equally effective protection against all the pathogen race without discrimination. And so far, we have got several studies have tried to map the APR loci in the oat genome, in which they use different biparent population and identify different QTL. And the marker linked to the QTL was converted into the CAT assay to support the selection for product resistance. So in this project, we tested advantage on the law list and then the material from this study. So we imported the source of resistance into Australia and tried to compare the resistance with the current germplasm in Australia. Here we can see that it is a result from an APR assay in glasshouse, compare the resistance between the imported material and the one the cultivar from Australia here. So you see that the one from imported material showing resistance, and so it was tested against the PCA isolates in Australia. So we see that the material also resistant in Australia. So there's an opportunity for us to try to integrate the resistance from this sort into the Australian genetic background. And then the second approach we'd like to take in this project is try to, instead of using single gene resistance, we would like to stack multiple gene QTL from different background into a preferred background. And with that, we can try to create multiple resistant layers in the plant. However, this is quite a lengthy process that requires multiple cycle crossing to integrate different gene, different QTL into one background. So the thing that we would like to use marker-assisted selection to support this process. And with that in mind, we have adopted a number of different published markers from literature, and then we testing against the Australian germplasm to see if the marker also specific for the resistant source here available in Australia. So that information will be used for the future breeding program. So now I'm going to talk about the two main parts of my presentation. The first one is the adoption of the Cas marker associated with crop rust resistance, and the second part is about examination of the oat difference set for crop rust. So what is the difference set and what is that used for? So it's a concept of race typing using the difference line. So for this, the race typing, also called the pathotyping, is a process to assign the phenotype code to the different isolates. So with that, we can identify what pathotype are present where in what frequency. So here I'm just want to show you a paper published by Chong in which he describes about a North American system of nomenclature for the PCA population. So in that paper, he used 16 single gene PC9 to study the frequency and distribution of the PCA population in different countries, including Canada, the United States, and Mexico. So here in the column is a number of different isolates was assigned to different IDs using the phenotype course. So how he assigned the ID to each isolate here in his study, I just want to give you an example here. So here's an isolate named DLPB. So how he assigned the code to that isolate. So each letter in the isolate was equal to the virulent profile of each PC gene subset here. So 16 PC single gene PC9 was defined to 4 subsets. And here for the letter D, it was assigned based on virulent profile of the PC gene subset 1, in which only the PC46 was ineffective. And for letter L here, so it was assigned based on the virulent profile of PC gene subset 2, in which only the PC38 was ineffective. And so on for the letter P, it was assigned based on the virulent profile of PC gene subset 3, in which 3 PC gene 51, 58, and 59 was ineffective. And so on the letter B, in which that was assigned based on the virulent profile of PC gene subset 4, and none of the gene was ineffective. So that was the race typing process. And nowadays in different countries, they use different set of difference line, and the reason for excluding or including the difference line in the set is based on the geographic difference in the virulent frequency, or based on if the line is important short of the for the resident in the breeding program or not. And here we are looking at the comparison of the two different sets, one from USA and one from Australia. There's 40 from the USA, and in this project we acquired the line from USDA, and there's 47 from Australia. We acquired from Australian Grain Gene Bank here. So comparison between two sets, we identified 29 in common as marked in the green color here. However, looking more closely to these two sets, both of them are very large, and for the purpose of phytotyping work, and some line in this set might not be effective at the moment, and not all share common genetic background, and some line with multiple genes, or some line carrying the same gene. So some of them have very poor genetic characterization. So all of this leading to the phytotyping problem, to the accuracy when you conduct the phytotyping experiment, and also the result of phytotyping for communication between groups. So in this project, we aim to reduce and sort out a common set for the phytotyping in Australia. And to do that, we first conduct the genotypic analysis in the different set, and the set was genotyped using the DASIC marker platform. So here's the result. We identify more than 65,000 markers across on the chromosome mapped to the Oats version 2 reference genome. So here the color coding based on the marker number per kilobase. So we identify a good amount of marker distributed across all the chromosome here. So with this good set of marker, we generated a maximum likelihood phylogenetic tree. And based on this tree, we can look at the genetic relationship within the difference line in the set. So here's the first thing we're looking at. We identify 3 different new groups stay very close to each other in the tree here. And that makes sense because here the new supposed to be sharing very similar genetic background and only different as the PC gene locus. So we have 3 different new groups: Pentecnear in yellow, Fraser in green, and Macru in blue here. And beside the new group, we also looking at the 29 common between both sets here. However, we highlighted that there, in color here, there's 8 paired lines that's supposed to be identical, but they are stay in different branch. So, and it just suggest that they are having different genetic background. And based on the genotypic discrepancy, we want to see if that discrepancy also converts into the difference in the phenotypes. So here we phenotype on the difference line using 48 PCA isolate to generate a virulent heat map. So here in the heat map, we have the color coding. The red as high virulent, the orange is the medium virulent, and yellow is the low virulence. So here looking at the virulent profile, we see that the lines supposed to be identical. They show substantial difference in the resonance profile. So just confirm what we found from the genotypic discrepancy here, and they suggest that they are derived from different resonance sources. And just zoom in one of the pair here, we have the PC70 different between the US set and the Australian set. And then interestingly, we see that the PC70 stay next to the PC39 from US set. So looking at the virulent profile of these two lines, we see that they share very similar resonance profile to the isolate. So this just indicates that the PC70 may be a replicate of the PC39 in the USA defense set. So this could be due to the issue with the cis mix-up because of the cis amplification or during the cis transportation process. And all of the findings that I have just shown you here have been published into paper online here. So if you want to look at the result in more detail, please go online and look at this paper. And now I'm going to talk about the second part of the project is that we try to adopt the gas marker for the literature and testing against the Australian germplasm. And in this slide, I plan to outline the approach to utilize the gas marker information in this project. So there are 4 different steps. The first one is we first test on the gas marker in the different set to see if any marker that's showing specific for the PCG carrier. And from that specific marker, we then screen the oat collection, including 115 line, to identify any other potential PCG carrier, which is already available in our germplasm. And then we will use the pedigree and the resonance profile to cross-check with the result from the genetic data to see if that makes sense. And here just quickly showing you a CAS genotyping platform that we use for this study, which is very high throughput for the experiment and the data analysis of the CAS marker. And due to the time frame of this seminar, I will just present the finding from the marker work for 2 PCG, PC91 and PC39. And for the PC39, so far, the virulence to the PC39 for PC91 in Australia already appeared in the last decade, but here we haven't identified the virulence in the Western Australia yet. And you know that Western Australia is one of the major growing oat, major growing region for oat in Australia for the milling industry. So it makes sense for us to try to deploy this PC91 in combination with the other effective gene to prevent an increase in virulence to the PC91. And to do that, we look at the literature and find out that there's a paper published by Nguyen in college in which he already identified a key gel linked to the PC91 location using 3 different biplant population, and the marker linked to the PC91 key gel was already converted into the CAS assay. So in this project, we're testing different marker and identify marker that's very specific for the PC91 carrier. So here is a CAS assay for the PC91. We have the line in purple are the lone carrier, high-5, amaclone, and PC91. So this line was already identified in this paper as well. And looking at the pedigree, the 3 line, amaclone, and PC91 and high-5, seem to inherit the resident PC91 resident from the Avena magla And actually the Hi5 was one of the parent in the mapping population for PC91 in this paper. And from the result here, we have the Hi5, which is homologous for the resident allele, and PC91 and alone was heterozygous for the resident allele and the susceptible allele. So this situation in the paper, they mentioned it was because there is a tandem application throughout the PC91 region. So here we also identify Volta as a heterozygous for the copy of the resident allele. And before Volta was postulated to carry two genes, PC50 and maybe additionally there's PC68. However, this first time Volta was identified as a PC91 carrier in Australia. And after testing the specific for the marker, we then using the marker to screen the old collection that include 115 lines. And I want to mention that in this set here, we have some lines such as Volta and Aladine. There's some line derived from different seed sort, like Volta 1, 2, and Aladine number 1 was derived from the AGG seed sort at the F6 and F7 generation. And we have Volta number 3 and Aladine 2 derived from breeder seed sort at F15 plus generation. And based on genetic result from Cas assay here, we identify drover as the carry the resident allele of this 91, which is homologous. And we have Aladine number 1 and Volta number 2 heterozygous for one copy of the resident allele. And in contrast, we have Aladine number 2 and Volta number 3. They are homologous for the susceptible allele. So it seems that the Aladine and Volta was still segregating at the early generation. And we also cross-check the result from genetic data with the pedigree. So here in the pedigree, we have the line in green here are the one that testing positive for the marker. And looking at the PC91 pedigree, we see that Drover, Aladine, and Volta, they only have PC91 in the pedigree. So that makes sense, the result from the genetic, from the Cas assay here, that this line carry PC91. And then we go on and check the resident profile to see if the result from the pedigree and the Cas assay matching with the phenotype as well. So here we're looking at the resident profile, including the PC91 carrier and the one positive for the marker, the one in green here. And we have the Drover and Volta and the Aladine having very similar resident profile as the other PC91 carrier here. And except that Aladine 1 here seems to carry additional resident based on the profile here, because that also resident to one of the isolate, which was virulent to the other PC91 carrier. And for the Volta number 2, testing positive the marker, and here looking at the resident profile, it's like they have the resident as combination of the PC91 and PC50 here. And looking at the pedigree, that makes sense because Volta actually also have PC50 in the pedigree. And then we looking at the Volta number 3 in the resident profile here. They're sharing very similar profile as a PC50, and it wasn't positive for the PC91 marker. So we deemed that this Volta number 3 may just carry only PC50 in the background. And we have Aladine number 2 here wasn't positive for the PC91 marker and having very different resident profile as the other PC91 carrier. However, we don't know what species gene contribute to the resident in this line. So on the result I have shown so far for the Cas work on PC91 just indicates an issue with the heterogeneous seed stock, and some of the line that have different version in the set may be segregated. So it's important for us to use Cas marker to track down the resident in the germplasm in the breeding program. And now I want to talk about some interesting finding from the marker work for PC PC39. And the marker for PC39 have been recently identified by Chow and published in the paper here, where they use two different populations to map the PC39 key tail. And then the marker was converted into the CAS assay and testing again a diversity panel, and they found it specific. So in this project, we adopt those markers and then testing again our Australian germplasm and to see if the marker also specific in our germplasm. And here's just briefly some information about the virulence to the PC39 in Australia. So you can see in the map, the virulence to PC39 already appear in Western Australia, but looking broadly, we see that PC39 seems still effective for more than half of the isolate in Australia. So it still makes sense for us to try to combine this PC gene with other effective PC gene to prevent the increase of virulence to that PC39. And here's it's showing the result, one of the most specific marker for PC39. And this is a marker testing against the different set. And based on the CAS assay result here, you see that it's successfully separate two different clusters. One is a resistant allele, and the other one is a susceptible allele in blue here. And the line in label here are the positive and negative control. And after that, we use a marker and then to screen for the old collection. And from that, we identify 24 positive line, in which 8 was lone carrier and 9 are potential carriers based on the pedigree, and 7 had no pedigree connection. So here's the pedigree tracking result. The pedigree was converted into the Helium map here. So looking at the map here, we have the line in green are the one that testing positive for the marker, and we also have the other line testing positive for the marker. In this study and also in the other study is presented by the mixture of the green and yellow here. And based on the pedigree tracking and the genotypic result, we want to see if the resident profile between the potential carriers also matching with the PC39 carrier or not. So we have the profile PC39 and Mitica. We have the Mitica here previously postulated to carry PC39 in the serial RAS report. And here looking at the resident profile, we see that the Mitica testing positive for the marker and also sharing very similar profile to PC39. So it just validates the finding from previous RAS report. However, we also have a number of other lines that in this study testing positive for the PC39 marker. However, they were never postulated to carry PC39 before. But looking at the resident profile here, so many of them showing similar resident profile as a PC39 carrier. Except that here we have Kuwa and Warigo look like they have some additional resident shots in the background because they show resident to couple isolate, which was virulent to PC39 carrier. And moreover, we have Liuxin and Ginny testing positive for the marker and having PC39 in the pedigree here. But looking at the virulent profile, they are different from the other PC39 carrier. And the fact that could be they sharing some similar in the genetic background around the PC39 region. However, the marker may be already been separated from the actual resident gene. So that means that the line here doesn't carry PC39 based on the resident profile. And we're looking at the glider here, testing positive for the marker, and they show very similar resident profile as PC39. But before this line was postulated to carry PC58 and PC59. However, looking at the virulent profile, they are different. So just suggest that glider actually carries PC39 instead. And the same for Warigo here. Before it was postulated to carry PC61, but in this study, we find out that it positive for PC39 and having similar resident profile as a PC39. So just to get this line actually carry PC39. Although gene wasn't seem to carry a PC39, but they also have different residence profile to PC61, which was postulated before. So we're not sure which PC gene this one is carried. And those were on the result from the study in the oat plant. And now I'd like to highlight some finding from the pathogen work and done by PhD student Eva Henningsen here. So in that study, she looking at the diversity of the PCA isolate in Australia, and the paper have been recently submitted on the preprint. So in that paper, she used the haplotype and pangenome approach to uncover the somatic hybridization recombination, and also she described about the intercontinental migration in the oat crowras. And I would like to show you some key figure from the paper. So here the first one showing you the diversity of the PCA isolate across 4 different countries: Australia, South Africa, Taiwan, and USA. So looking at on the top here is a result for the phylogenetic tree for the USA PCA isolate. They look very diverse and look like there's multiple different lineages present in USA. And compared to the PCA population from South Africa and Taiwan, here look like they are mostly from clonal evolution. And part of the PCA population from Australia also seem to be clonal as well. However, from the recent collection of the PCA, we also identify a quite diverse population of PCA, in which that's separate into 18 lineages. So looking at the map here of the lineages by state here, we have the Western Australia, where there is the most diverse of the diversity of the PCA population. However, actually the diversion in the xenotypic wasn't converting into the phenotypic diversion. So in that paper, she mentioned about the masking effect of the one haplotype on the other haplotype in the PCA genome. So if you want to look for more information about the paper, please go online looking at that preprint. And now I'd like to conclude about the finding in this project. So far, we would like to seek some correlation in the community to try to standardize a difference set that could be used for pathotyping. And the second thing is that we have adopted a number of different markers, which is diagnostic, and have been able to identify several potential carriers. And the third is that we have used approach of using molecular marker, pedigree, and resident phenotype to characterize the outline, and we have checked out resident source in the plasm. And the result from this project have provided some valuable tool for the assist the selection of oat variety with stacked crowras resident gene. And some future work in this project will be that we will keep adopting some Cas marker linked to the API loci and testing again our Australian germplasm to identify the potential carrier. And then we will conduct isolation analysis to identify the API loci in our germplasm and environment. And we will also develop some population for API mapping and creating the material for breeding. And yeah, so at the end, I would like to acknowledge all the people contribute to this project, and also acknowledge the people contribute to the germplasm, and also the people donate the red sample to our project. And that's it for now. Thank you everyone for listening to my talk, and then I'm happy to take any question regarding this presentation.
Thank you so much, Duong. That was a really fascinating talk. You've done some amazing work, you and all the people there at CSIRO and collaborators, of course. And Yeah, the whole, I mean, pathology, it's a very complicated thing. I often, even though I've worked with molecular markers for, crown rust and stem rust and things, it's such a complicated thing, and I certainly can't claim to understand even very much about it necessarily. We do have one question, or we did have, it's sort of disappeared. I'll get to that in a second. And yes, we're very rusty doing these webinars. So yeah, got to get back on track here. Because one of the things I wanted to mention, actually, you mentioned James Chong's work, and he's actually on the call. So James, I don't know if you have anything in particular to add to the conversation, but yes, your work, of course, continues to be valuable. And Mike McMullen is also on the call. And of course, he's the one who bred the line Hi-Fi, so that has PC91. So they are actually both here, and I'm sure appreciate the work that you're doing too. So I had seen a question here. I guess, it was answered. However, not everybody knows that it was there. So Lydia Bitz was wondering whether Amegalon and Hi-Fi that you were using are the same ones that were used in Panout in that research. And Melania has said you guys didn't actually provide the seed for that. So it is quite possible that they're different sources and they're not exactly the same. And that, of course, is one of the really big issues in all of this kind of work, as you've pointed out so well here, is that there are a lot of differences in lines, whether, seed mix-ups or segregating populations, various things, and of course, genetic drift, all of that type of thing. So the floor, as this thing, is open for questions. So if people would like to ask other questions, that would be great. I do have another sort of comment / question. So I know a few times I had asked people, we have all these sets of differential lines, and I didn't until recently realize that there were sort of two sets that the community was using. But I'd often wondered why they weren't in the gene banks. And of course now, I understand more that they're just too variable. There are too many issues with the lines, and because some of them are less well characterized, all the things that you said in your presentation, that there are sort of problems with some of those. And so I'm wondering if going forward, one of the things you might be doing is perhaps working with the gene banks and so on, as well as the Rust community as a whole, and to try to better characterize these lines and then have them deposited, say, in the gene banks where more people could access them. Do you think that's going to be happening?
Yeah, thank you for the question, Charlie. I think it is, that is good suggestion that we should try to work closely between group and with gene bank to try to have a common set for all the different group to use the same thing. However, I think it's a pretty hard process, and I think Melania have been trying to talk to the other group in US and try to talking about the idea to create a setup like the new like I mentioned in the phylogenetic tree, we should have all the PC gene that stay in the same genetic background and only different for the PC gene local. That will be the most useful for us. Yeah, because who know? I mean, what happened if if we have the different PC gene in different background? Who know what else in the background? And and to collaborate with the gene bank, that's a difficult process. I think Melania we know more about that. Yeah. I think she raised a hand here, so
and she
may
has
comment
raised
on that.
her hand. Yeah, so go
Yeah.
Yes, yes, thank you, thank you, Charlene. Yeah, the deposit the seed in the gene bank here in Australia is something that was discussed with GRDC. It is the trouble. We could do that, but I think it would need some constant checking from the community that we are keeping the stocks basically intact. I think with oat we have the difficulty that it is an outcrossing species. Is more complicated than keeping up with the wheat differential sets. So yes, it's not an easy task, but we have had conversations with some of the key labs distributing some of the seeds. So, and there's also efforts from other groups in the US and Canada in creating a set that is more in a common genetic background, so that could ease some of these problems. And because I have my colleagues here from Canada, we didn't have time to include this in the presentation because it starts getting very repetitive. But for instance, a stainless that is supposed to be carried PC91, it was not positive for the marker. And in fact, the virulence, that the resistance profile is quite different than what it should be. So we don't have the right stainless. So we are currently, I don't know what we have, but it's not it. So we're kicked it out from the differential set that we have. We're not using it anymore because we just don't know what it contains. But if someone from Canada could help me getting the right stock, that would be very helpful. And yeah, so I think it's about trying to integrate as many data sources as Nguyen was explaining. The pedigree, certainly the resistance profiles, the pathotypes, and the markers, and being conscious that we don't have enough markers for every gene that has been described. So there's still a lot of work to do there. But yes, it would be great if we could engage with the community and try to work together in sorting out some of these issues. And it is difficult because some of these genes already have been overcome. So, the funding bodies will say, well, why do we want to invest on characterization of a gene that was already defeated? But I think the justification is that some of these genes, as we have published recently, may be represented in the differential set as alleles of the same gene or duplicates. So I think there's value in doing that characterization and then learning what is that is not useful in the breeding pipelines, and then we can discard it because there is there could be a masking effect from these other genes as well. So but yeah, so that's where our thoughts are.
Right. Yeah, no, I think that's very true. And if I remember correctly, doesn't PC38 act as a repressor? Oh gosh, we're gonna have to go look that up. But yeah, and sometimes I think, right, because I did do some work on PC38, and the idea is too that you're not necessarily selecting for something. Occasionally you are selecting against something, and so
Yes.
you need markers for those types of things.
Yeah, and the other results that we have is that we're very comfortable in making assumptions of genetic relationships of isolates based on the pathotypes that we are describing. And I think there we have to be very careful because, like you said, Charlene, we have inhibitors in the host, and also we have inhibitors in the pathogen. So although we would like to think that is a one-on-one relationship between this AVR effector and the immunoreceptor in the plant, is more complicated than that. And when we overlap the genetic relationships that we can extrapolate using whole genome sequencing, so that's what is closest to the truth, to the biological truth, right? And then we compare to what we could be saying with pathotypes, we start seeing the discrepancies and the gaps, and basically, where we could be misled if we only consider the phenotypes.
Right.
And the other part that is also important to consider in that area is that, the rust have two separate nuclei that we know now they are basically moving independently. So they could they could have these genetic exchanges where one nucleus is donated to another strain. And so we should be thinking more about the content of those haplotypes. And that was the idea behind going to this pangenome, which we now have 9 isolates characterized. So that both haplotypes of those strains for Australia and 6 for the US. And we're still growing that resource with some isolates from different countries. So, and we started actually seeing that movement of the nuclear haplotypes from the US to the UK. It's a very interesting phenomenon, but I think what tells us is that we need to be thinking more about what is present in those haplotypes as opposed to just considering both together because of that masking effect when you have dominance of a virulence, right?
Mhm.
Which is essentially what we had here in Australia with wheat stem rust. So the most predominant lineage of wheat stem rust here actually has half of the material from UG99. And it's been here since the 1950s, but we had no mechanism to detect it until we resolve those haplotypes of PGT21-0. So similar situations could be happening also with oat crown rust.
Right. And we tend to focus, well, as a community, tend to focus more on the plant side rather than the fungus side. But I mean, yes, you're totally correct. We really need to understand what's going on the fungal side as well. And yeah, those 2 nuclei, that's it's a little bit tricky to sort all of that out for sure. One other thing too, I was thinking of for some of these genes, again, if somebody's looking for funding to support some of this type of work, I know people, because we did it here in Ottawa for a time, we've been recycling different genes too. So after a while, because the races of rust and so on have changed, you can actually bring back in some of the genes that had been previously defeated, and they will work again. So that could be another reason, instead if people are looking for funding, ways to address those issues when they're looking for funding.
Yes, that's a good point.
So, and we do have another question from Edita Paxos. Gosh, I'm trying to learn Polish, and I'm sorry, Edita, I've messed up your last name. Paxos, I think, perhaps. You can correct me, I'm probably wrong. Her question is, have you checked if the Cas marker for PC39 was amplified in PC70?
Oh, yeah. Yeah, thanks for the question. And I can't remember exactly on top of my mind, but I can answer you later because I do have to read now. But I will tell you if the PC39 allele actually present in the PC70 or not. But I can send you answer later for sure. Yeah.
Can I add to that? I can't remember the top of my head, but we have to remember that we have 2 sources of PC70. The one that came from Australia from the bank, which seems to be different than the one we have in the US. So the one in the US is clustering, it seems identical to PC39. So that's where it would make sense to check for the marker. Not the one in Australia is quite divergent if you see it in the phylogenetic tree. But the other point I want to make is that when we conducted the GWAS in the pathogen to map virulence associated to PC39 and PC70. So if you indeed have these specificities, right, they should be mapping in different regions unless the 2 AVR loci are linked. And in fact, they were in the same chromosome and in the same genomic interval, which is further support that the PC39 and PC70 lines in the US differential set are the same, or yeah. Does it make sense? So you conduct a GWAS, but instead of using the plant, you're doing it with the pathogen. And if they're different, they should be landing in different regions, unless you have a cluster of aviary infections, which is also possible. But that's another piece of evidence that PC39 and PC70 were mapping in the same area of the genome in the pathogen.
Right.
In both haplotypes.
And there are the clusters of markers, of course, or markers, I'm sorry, rust resistance genes, of course. I'm trying to remember, because I mean, I did just put together this big gene inventory. PC71 is on the same chromosome as PC39, if I remember correctly, but not in the same place. And not remembering about PC70. But sometimes,
yeah, but this is
sorry,
this is Charlie. This is going to the pathogen side, so they could be
right.
the resistance genes could be in different places in the plant.
Yeah.
So I mean, different scenarios, right? You could have different resistance genes that perhaps are recognizing the same effector, or because they are alleles,
right.
they could have very similar, recognition. And those that AVI effector then it would map in the same region of the genome of the pathogen.
You're right. Sorry. Yes.
Yeah.
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 Right.
Yeah. It's very convoluted,
I
right?
need to remember which side we're talking about, the plant or the
yeah.
So yeah, both at war. Is it right? Exactly. So yeah, because one of the things too, actually, just thinking, I'm too stuck on the plant side. I need to learn a lot more about the fungus side, but well, not fungicides, fungicide and fungicides, but the I suppose with a lot of these markers too, I mean, that is also a bit of an issue if you have a cluster, because I mean, how close do you think we are to actually identifying the genes themselves, like the actual, resistance genes in the plants? Do you think we are particularly close to that?
Well, yeah, I mean, we could adopt a lot of the techniques that we have in wheat. The problem is that we haven't had a really like that much support to clone the genes. I think it's not, we're not that far if we really put the effort into it. Some of these markers, I think they're quite close. They may be very close to the gene, others not so much.
Right.
But I think there was a talk someone told, I wasn't at the Plant and Animal Genome Conference in San Diego this year, but I think someone told me that they may have already one of these PC genes, or they may, I can't remember which group, but yeah, so it's possible that one of these will come to light very soon.
Because I don't know, I remember many years ago when I sort of started working in oat genomics, I was kind of wondering, well, if we could clone one of the genes, perhaps we could even make artificial resistance genes, by tweaking them somehow. But that, I suppose, would also be a fairly long way off. But yeah, there's so much to learn. There's still so much to find out. Does anyone else have any questions here or comments or things to add? I'm just looking at our time here. We are pretty much out of time. Going once, going twice. I guess if that's it, and it said we are pretty close to time, I'd like to thank Duong and Melania both so much for the presentation. That was really fascinating, and yes, illustrates quite well my lack of knowledge in certain subjects, but it's such a complicated thing. And Pablo, do you have anything to add? Perhaps I might have one thing just to, oh, hang on, there's another, wait. Oh, Lydia has a comment. Can you type it, or can we let Lydia speak? Which way would you like to do that, Lydia? She can speak. Okay, Pablo, can we? And how do we do that? There's a way.
Let me see.
You have that authority, I think. I don't seem to have that permission. Oh, there, I think that worked. Lydia, yeah, go ahead, please, Lydia.
Hi.
So,
Uh, hi, everybody. I don't know how to turn my camera on.
oh,
It's a new Zoom outfit here for me. I want to say, send the greetings from the north of Europe to all of you. And we are just about to start a new Nordic project. It's called Robot. We want to work on more robust oats for the coming changes, environmental changes that we can already see. For example, we are starting to have drought problems. That has never been case before because a lot of snow, a lot of rain. However, one of the issue in this project would be ground rust. It's not a problem at all yet, but it seems that it's starting to be as well here. And like on the first slide of the presentation, that was nicely visible that north of Europe is not suffering from the crown rust. We are struggling a lot with Fusarium, but this is coming. And we are planning to test Nordic core sets of oats to try to find differential sets. And also we are going to try to study the pathogen itself. So what kind of races do we have here? I'm not the one particularly responsible for that. Maria Jali is the PI. I am helping her to co-lead the project. And there are other colleagues who are responsible for these tasks. But it would be great to start networking on this issue. And I'm really, really happy that I was listening, although it's here now past midnight. So that's it.
great. Thanks so much, Lydia. Yes, and thank you for joining us because I know, yes, for you, it is extremely late. And of course, it's extremely early for Duong and Melania. So thank you so
Yes.
much for fitting everything in. And Pablo, do you have anything to add then?
No, I would like to thank everybody for attending the webinar. And a reminder, if you have research or work related to oats that want to share with the oat community, please don't hesitate to contact either Charlene or me, and we can schedule a webinar so we can keep this Speaking of Oat going and keep the community connected. So thank you so much again.
Thank you. And here's there, I'll put up this slide. There we go. TBA, the next one. But yes, please, if somebody wants to speak, get in touch with one of us.
Thank you, Charlene, and thank you, Pablo.
Pablo.
No, thank you so much. It was a great seminar and it was nice to meeting you, Duong.
Thank you.
I apologize that my camera would not turn on, so it's just the mysteries of computers, but at least I could talk.
Yes, maybe you're on the same system as Lydia. You know,
Yeah.
she had trouble too.
All right.
Thanks again.
Thank you so much.
And bye-bye. Yeah.
Take care. Bye-bye.
Bye now.